He has served as editor and assistant editor of few journals and also as an expert of IUCN Red List assessment of orchids for the state of Arunachal Pradesh. Mr. Arjun has published research articles, book chapters, and many science articles. He also has several poster presentations at national and international platforms. As he is the tip of the iceberg, and an illustrious career is there to follow. Without taking any further time, I welcome Mr. Arjun Adit to deliver his talk on plant taxonomy through the lens.
Thank you so much, ma'am. It is an honor to be here and speak among students and teachers. I'll just try to share my screen. Uh, is my screen visible?
Yes, yes, it is visible.
Um I would like to take this opportunity and thank all the teachers and organizers in Shivaji College and Department of Biotechnology for funding the program, which I hope will help all the students and participants in the program. Thank you for your words, ma'am. I'm deeply honored. This talk is more or less going to be interactive at first, and in the later stages, I'd like to present one or two case studies, and then end up my talk with questions and answers. I'm sorry, we are facing some network connection problems in the department today. Uh, I'm also sitting in the uh, tissue culture room to catch my network on my phone. I'm using my hotspot. So I may break up, my voice may break up. Uh, I urge the organizers to intimate me, and I can repeat myself whenever required. Uh, I want the attention of all the students who are participating. Most of what I'm going to show you may already know and maybe a revision for you. Uh, I'm aware that you have already had lessons on taxonomy and microscopy earlier this week and in the last week. So it is going to be a interactive quiz session in the starting. Uh, am I audible? Still audible?
Yes, yes, you are audible, but I think they don't have the rights to unmute themselves. So
Okay.
probably if whenever you are asking something, they can write in the chat box, all the participants, and then as you will not be able to see the screen that time, the chat box, so we can probably tell if any answer comes. I hope that's okay.
Yeah. That will be fine.
Oh,
So let's just start by saying that why do a taxonomist need to burn his eyes by sitting on a microscope? So basically, taxonomy is a practice of classification which is based on shared characteristics. These traits, they are the defining feature of taxonomy. And the alpha taxonomy relies on microscopic techniques to identify features that are indistinguishable to human eyes. And the accurate analysis relies on specifics of shape, texture, and aspect ratio of major or minor structures. So more than often, there are these three types of microscopes which are uh, majorly used in the taxonomic studies. What do students think this machine in the middle is? What kind of microscope do you think this is at the center?
participants, you can write in the chat. Guess one. Kachuk has written
Achuk
SEM.
has written.
Same. Yes.
Yes.
So that is right. This is the scanning electron microscope. What do you think on the what do you think this microscope on the left is? Anyone? Have you seen this? I'm sure you must have this in your college.
Yes,
Okay, Kajal
Kajal.
is Kachuk is
Yeah.
also written dissecting microscope.
Yes, this is the dissecting microscope or the stereo microscope. Uh, Basically, the structural analysis of features is usually done using these two microscopes. But we have a third one which is used in the anatomical analysis or anatomical discoveries. What do you think this is? The one on the right.
Okay. They're writing compound.
Uh, yes,
L-
this is a compound microscope, but more commonly this is known as a
Electric
binocular,
Electric
compound. Or
yeah, a
light
yes.
binocular or a
compound.
bright field microscope. So we have these three types of microscopes, which are more or less more frequently used than others. The other ones are transmission electron microscope um, and the fluorescence microscope, but they are not those are that oftenly used in taxonomic studies. So one of the major thrust areas uh, in the use of microscope is the palinotaxonomy studies. These are the studies of pollen structures, and as you can see from this slide, this is a scanning electro micrograph, but this is pseudo colored. So the electron micrograph are usually black and white uh, in their output, but pseudo colors can be used to help them distinguish better. So, students, I don't know how much of you have had taxonomic lessons in your bachelor studies, but it would be wonderful if any of you could guess uh, what plant family this pollen grain belongs to. So I'll just give you a hint. This is a monoporate pollen grain. It has just one single pore, and this is a characteristic feature of a plant family. Any answers to this? Any tries? There are no marks which are going to be deducted for this. This is just an interactive session, and
Yes. One student has written Graminy.
yes, OAC, Gramini or OAC is the correct answer. The family has monoporate pollen grains. What about this? Do note the spiny nature of the enzyme in this figure. I'm sure you are
Utkarsh
aware of
has
this.
written Malvasi.
Uh, that is so close, but that is not the right answer.
One has written Asteraceae,
Yes.
Geetanjali.
See, one of the major differences between Asteraceae and Malvaceae is that Asteraceae has uh, the exine of Asteraceae members are spiny in nature as compared to the Malvaceae pollen grains, which are not those sharp, and these are known as tuberculate or club-shaped pattern of exile. So the Asteraceae members have sharp spines as compared to Malvaceae members, which are usually having the club-shaped spines or blunt spines. See, these are the examples of how scanning electron micrographs are useful in distinguishing in identifying the different taxa. What about this? This is a pollen dyad. In many cases, the pollen grain is not released in form of monads, as seen in previous figures. Okay, I'll just go on and say what this is. This is pollen diodes are usually a characteristics of some aquatic families, one of them being Podostemaceae, to which this belongs to. What about this? These are very common plants that you always see around you. I'll just give you a hint. Uh, they belong to Amaranthaceae. The one on the right and one on the left are different genuses, but in the same family. You can see how diverse the structuring of pollen grains in the same family, how they differ in the same family. While I wait for the answer for the one on the left, this is the Amaranthaceae family, but the one on the left, I would like to know a genus for it. And while we wait for it, this is an example of, and this is commonly used not just in taxonomy, these kind of techniques, but they are also used in uh, identifying the pollen allergens. So usually hospitals they employ uh, skilled uh, microbiologists and skilled microscopy technicians to identify the more occurring uh, pollen grains around the area, because most of the pollen grains can cause allergic concussions or allergic problems or asthma attacks to people who are prone to bronchital problems. And what they do is, yeah, was there someone answering some question?
No, no. Actually, Arjun, we have allowed the participants to unmute because
Okay.
this did not sound like a quiz.
Okay.
Yeah.
If they answer themselves, it will be a better, experience for you also.
Okay.
So we have allowed them to unmute. But kindly, please, whenever you are answering, then only unmute. Don't disturb the speaker otherwise. So if you're asking any question, now you can ask and they will directly answer to you. Okay, Arjun?
Sure.
And I think Aman has written alligator weeds.
No, but the one on the left is Amaranthus pollen grain. Uh, Students in North India might recognize it using the name cholai, cholai ke laddu, which are very famous. So these are the pollen grains of that plant species. And so I was talking about how these same images of pollen grains are helpful in understanding the allergic problems that people have. So usually, what hospitals do, they they take a glass slide and they make a film of glycerine on top of it, and they just hang it around in different parts of their district or different parts of the area where they want to understand what kind of pollen allergens occur. So over a course of a day or two, a lot of pollen grains stick on this glycerine slide, and they are then observed uh, under the scanning electron microscope post the critical point time, and then they identify what kind of pollen grains are present in their vicinity. So this type of study is known as maintaining a pollen calendar, because this exercise is done uh, in different seasons, and then people get to know what kind of pollen grains occur in different seasons and what kind of pollen grains uh, they they are around in the air, which of them are more frequent, more abundant, and uh, more likely to cause allergic reactions. So moving on, uh, any ideas on what this structure is?
Utkarsh, please unmute and answer yourself because you've written rightly.
Calotropis pollinia.
Yes. So this is a Calotropis pollinia, and there are very few families in the plant kingdom which have the Pollinia or Pollinaria sacs. And Asclepiadaceae, now a member of Epocynaceae, is one of those clades which has a characteristic feature of presence of pollinia instead of pollen grains. This is one other plant family, the Orchidaceae family, which has the pollen grains uh, inside the pollineal sacs. These all belong to different genera. The one on the top left is from Acampé. The one on right is from Papilionanthe, and uh, the third one at the end is from Cymbidium. So even though we have the pollinia, just like pollen grains, they have different morphology, and based on their different morphologies, people can identify different species. Uh, what do you think is the difference between pollinia and pollinaria? I'm sure most of you must have read this again and again in test books, but in bachelor's, I don't know if you have had this question in your mind, but I certainly did. We have somewhere people write pollinia, then in other places people write pollinaria. What is this? What is the difference between these two? Can anyone just make a guess?
Sir, is it singular and plural form?
Yeah.
Pollinia is singular and pollinaria is plural, I think.
No, that is not right. But I think you might be going in a correct direction. So I'll just answer your question, answer my own question. A pollinaria is a unit, is the whole unit, which is a pollinating unit. So basically, if an insect arrives and the whole structure sticks on the insect's back, that is pollinaria. But the unit which is embedded into the stigma is pollinia. And what are the main differences? The main difference is that this this region on the bottom on all three these three pollinia is known as the visidium, and this is the structure which sticks on the bee's back but does not deposit itself inside the stigma. And the loss of this structure, the visidium, is makes the pollinia, because the pollinia then has only a stipe and the pollen sacs. So the main difference between pollinia and pollinaria is that pollinaria is a pollinating structure, but pollinia is the depositing structure. And there is loss of visidium when we move from pollinaria to pollinia. So these are stereo microscope images of different species of Barlaria. Uh, you might know it from its common name Vajradanti. So as you can see that we have different uh, fruits, different shapes of fruits in different species of Barlaria. Even the seeds are quite different. And this detail, instead of being, scrutinized under the scanning electron microscope, is quite visible in the stereo microscope or dissecting microscope, and one can just see different features. See these thorn or horn-like structures inside the pods of Barlaria help in dispersal of the seed. These thorns, not just the shape of the seeds and the fruits are different in different species of Barlaria, but also the shape of these thorns and the length of these thorns. Some species they accommodate more than uh, 3 or 4 seeds inside their pod. Some like Barlaria prionitis in the third figure in the bottom left, they are able to accommodate only 1 or 2 seeds inside. And you know, not just well, you if you move on in plant studies, you experience a lot of stuff, and you get to know that it is not just the flower which can help you in identification of a particular plant species. It may even end up being a fruit, and this is a prime example. So in other sets of studies, people may try to look at the trichomes, and this is a labellum of Cymbidium species, and at inside the same petal, inside the same labellum, we have 3 regions where 3 different types of trichomes are present. The first, the green box, we have it as B, where the trichomes are in form of small conical cells. The second red box labeled as C are longer trichomes, and the blue box labeled as D has other emitting trichomes. I'll just elaborate what this is. So basically, as we had moved on and tried to study different orchid species, one of them being Cymbidium, we saw that uh, these 3 types of trichomes on the same plant, on the same flower, had different uses. So the bee usually lands in this box, near this box, where near this green box, where small conical cells are present. These conical cells help in adherence of the bee so that it can just wait and sit on the lip. The longer trichomes labeled in the red box are osmophores, which chemical compounds that help in attraction of the bee. So these trichomes they release a particular kind of scent which attract the bee, and then the bee comes back to the flower and helps in pollination. The blue box, uh, we have trichomes which are known as eliophores. So these eliophores secrete lipid. So not in all cases the bee goes for nectar. In many cases we have minor rewards where the bee might go for uh, rewards other than pollen or nectar. So we have here in this species we had lipid as a reward, and these eliophores they secrete lipids, which is the main reward for the bee. So you can see the bee comes and sits near this green box, is attracted by the red box, and is rewarded with the blue box. So these kind of studies also help in taxonomic identifications, in enumerations. Later on, in different species of Cymbidium, we might see different sets of trichomes, which can be later characterized, and then a phylogenetic tree based on the characters itself can be made. So, moving on, I would like to share a small story with you. So, there's a big genus in Orchidaceae. It's known as the Oberonia or fairy orchid. It's an epiphytic genus which is mostly found in tropical areas. And as you can see from this image on top left, the size of uh, the flower is quite small. So you have a scale of 5 centimeter. Just imagine how small these flowers on this inflorescence is. And based on the uh, different characters on the petal, most of these species have been delineated. And because this genus is so morphologically plastic, uh, there are you know in many instances they have caused problems for taxonomic studies, for taxonomists to identify, to delineate whether two species are different, whether a species is new to science, or whether that is a synonym for something else. So it's been a dilemma for many orchid enthusiasts. And as you can see from the slide on the right, the figure on the right, these arrows mark the gaps which are consistent for one species. So these small gaps can only be studied under the scanning electron microscope. And even stereo microscope or dissecting microscope does not give a clear image or understanding of these differences in different species of Oberonia. Many students and many young botanists, uh, they have a thought that different colors or small slight variations in flower color or flower shape or flower structure or texture may be due to them being different plant species, but that is not true. All three images in this case are the Oberonia mucronata, which is the same species. And as I told you before, see this area at the bottom where this gap is present, and this is consistent in both these orange and green flowers, and that is how people could make out that this is same species, Oberonia mucronata. But not always do we find new species, or do we match that two things are same or not. In many cases, it is a discovery in itself whether a species which was earlier defined is a synonym of something else or not. And here again is a prime example. So these first two figures A and B, they were earlier plant species which were known as Oberonia titania, but then C and D were considered new species and they were classified as Oberonia mucronata. of fossil plants where you find new species, you need to classify them, and you have to take the aid of stereo microscope and scanning electron microscope to classify them, to put them in their right places. In another case, uh, these pollen grains, these pollinia, helped in you know correctly identifying when Orchidaceae originated between the angiosperms. So we do know that this was always a question: which of the plant families is more advanced, or which was later evolved, or which is more recent or recently evolved. And there's always been a debate between Asteraceae and Orchidaceae. And even though sometimes people say that Asteraceae is a dicot and dicots have come later, and Orchidaceae is a monocot, and monocots had diverged earlier, then how come there could be a clash between Orchidaceae and Asteraceae? But you have to understand that you are looking at a dendrogram, and when you compare the dendrogram in terms of time scale, you understand that there are some families which have evolved later. And here in this case, since this orchid, this pollinia of an orchid was found on a stingless bee in which was stuck in amber. This when this pollen grain was analyzed, it was put into not just a new species but in a new genera, Meliorchis caribae. But then this structure was carbon dated, and it was discovered that this belonged to the late Cretaceous period. And based on this, observation, uh, not just a new genre was defined, but also the origins of Orchidaceae as a family, could be established, clearing the more or always elusive question of which family later came, the Orchidaceae or the Asteraceae, and Orchidaceae came out as an answer. And when uh, these characters for so many clades are analyzed, people understand by understand people trying to understand the evolution of such characters can place them on the phylogenetic tree and look how uh, different characters have evolved over the plant families. This is just a small example where we try to trace the characters of nectaries, and we found that there has been a nectary shift, the shift in nectary position from uh, outwards of the flower to inward in the flower. That is, in the earlier clades or the Anna clade, we had uh, we had nectaries which were on petals and sepals, which later went on to move inside the anthers, base of the anthers, uh, on the base of the ovaries, and then towards style. So we had these nectary states which we understood, and which we could conclude to say that nectary as an organ moved from outside the flower to inside the flower in an acrocentripetal fashion. So microscopy, as we just saw, not just helps in uh, understanding the taxonomic relations between new species, delineating two new species, merging two species into one, um, identifying them for medical purposes, but can also be used for tracing the evolutionary history of different plant organs. And uh, as we had moved on in our as we had moved on to our, I'm sorry, I'm having a call. I'll just connect back again. Okay, I was just intimated that maybe I should have also used Hindi somewhere. So I was just summarizing in my last slide, saying ये बताने में कि टैक्सोनमी सिर्फ टैक्सोनमी एक छोटी चीज नहीं है और माइक्रोस्कोपी एक टूल नहीं है. मतलब सिर्फ टैक्सोनमी के लिए एक टूल नहीं है. माइक्रोस्कोपी आज के टाइम में मेडिकल पर्पसेस के लिए तो यूज हुआ ही है, जैसे कि मैंने पहले बताया, पॉलन ग्रेन्स देखने में जो एलर्जेंस की तरह कॉज करते हैं, या फिर दो स्पीशीज को एक बनाने में, या एक नई स्पीशीज बताने में, या ढूंढने में, बट ये भी बताने में, ये नए कैरेक्टर्स निकालने में और उनको समझने में कि उनका एवोल्यूशन कैसा हुआ है. तो बेसिकली लास्ट स्लाइड में मैंने ये बताने की कोशिश की थी कि हमने नेक्ट्री स्ट्रक्चर्स डिफरेंट प्लांट फैमिलीज में हमने ट्रेस किए एक फाइलोजेनेटिक ट्री पे, और हमने ये देखा कि जो इनिशियल जो अर्ली ब्रांचिंग प्लेट्स हैं उनमें जो नेक्ट्री है वो हमको पेटल्स और सेपल्स पे मिली, और जैसे जैसे हम अंदर आते गए फाइलोजेनेटिक ट्री के, एंजियोस्पर्म के फाइलोजेनेटिक ट्री पे, और जैसे जैसे हम लेटर ब्रांचिंग क्लेड्स में गए, हमने देखा कि नेक्ट्री जो है धीरे धीरे गाइनीशियम के नीचे आ गई. पहले एंड्रीशियम के नीचे, फिर गाइनीशियम के नीचे, पहले एंड्रोशियम और गाइनीशियम के बीच में, पहले पेटल, उससे भी पहले पेटल और एंड्रोशियम के बीच में. तो हमने ये कंक्लूड किया कि नेक्ट्री का जो इवोल्यूशन हुआ है एंजियोस्पर्म्स में, वो बाहर से अंदर की तरफ हुआ है. इस मूवमेंट को एक्रोसेंट्रिपिटल मूवमेंट कहते हैं. तो ये जो स्टडी हुई है, ये जो हमने कंक्लूजन बनाया है, ये एक रिजल्ट है माइक्रोस्कोपी का. कैसे? बाय लुकिंग, ये देख के कि नेक्ट्री कहाँ पे प्रेजेंट है हर क्लेड में. तो माइक्रोस्कोपी इतना इम्पोर्टेंट, इतना पावरफुल टूल है कि वो चीजों की एवोल्यूशन तक को बता सकता है. और अब फ्यूचर में लेटेस्ट जो इलेक्ट्रॉन माइक्रोस्कोप डेवलप हुआ है वो है स्कैनिंग हीलियम माइक्रोस्कोप. और बेसिकली इसमें क्या है कि जो इसका जो एप्लीकेशन है वो इतना तगड़ा है, इतना इम्पोर्टेंट है कि आप, जैसे ये एक एग्जांपल है, ये रैपिडोप्सिस का प्लांट है, इस पे ट्राइकोम्स होते हैं. तो ट्राइकोम्स की जो लेंथ है सिर्फ वो मेजर नहीं करता, वो उसके बाद में उसका एक मॉडल उठा लेता है और ये मॉडल उठा के वो जो है मर्ज कर सकता है किसी और प्लांट के साथ में और देख सकता है कि कितनी % सिमिलैरिटी है। तो ये अराबिडोप्सिस थालियाना है। अगर इसका कोई डिस्टेंट या क्लोजर रिलेटिव हो, उसमें जो ट्राइकोम्स होते हैं वो इससे कैसे डिफरेंट है, वो उसके ऊपर सुपर इम्पोज कर सकता है, मॉडल कर सकता है। सो दिस इज द लेटेस्ट technique of microscopy which is being employed in taxonomy. I would like to finish up here. I've not made a thank you slide, but just this slide, that is because I want people to focus. मैं चाहता हूँ लोग इसे देखें और सोचें कि फ्यूचर में कैसे और उनके आसपास कैसे माइक्रोस्कोप जो है इम्पोर्टेंट हो सकता है, कैसे चेंज कर सकता है कुछ. I'm open to any questions. I'm sorry, I'm
No,
sorry,
no,
to organizers, that I had not realized whether I should be speaking in a bilingual fashion. I hope no inconveniences have been caused.
no, Arjun, it was wonderful. And I really thank you so much for such an interactive session, which we didn't have before this. Your talk has made us realize how interesting the world of taxonomy is when viewed under microscopes. We are rather grateful that you could share a part of your research of last few years with us. Yes, there were a few glitches that is on part of ours job. So this is a earnest request to all the participants. From now onwards, if you have anything to tell us, please mention it in the Telegram. Please don't disturb the speaker. It is very difficult for a speaker to find out your chats like this. One person is asking for English, then the second person is asking for Hindi, and again going back to English. It's very difficult. Please try to understand. Your speakers have taken out very precious times from their schedule to actually give you inputs like this. And before I go ahead, yes, there is a question answer round, Arjun. So I will just take up. I think there were a few questions I could find.
Yes, and I will also like to say this to all the participants because we are from University of Delhi and our medium is English here. So even if we use bilingual mode and under explaining an answer, usually we prefer only English to teach because of that, so that every student coming from different backgrounds they also understand. And अगर आपको कुछ समझ में नहीं आ रहा है, please आप उस portion में ज़रूर पूछ सकते हो. That you're always free to do that. But and as far as I think, since 1.5 weeks we have been teaching in English only. So
That's what I
why?
was saying, that all the sessions have been in English. So why this session was pinpointed, I want to ask, means if
Yes.
at all we are creating any kind of problem for you, please inform us beforehand on Telegram. We want to help you out. But
So,
if there is
because
some,
especially we are calling people who are having expertise in some area just to give you exposure. Now, sir is actually doing PhD right now, so and he is into this sort of research. So we could only explain you things but right now with his practical experience he showed you some differences of at the family how you are applying whatever you studied in these 2 weeks, how you actually apply that in research. So I will appreciate, and not I only, all the organizers will appreciate, as ma'am said, if some issue like this is occurring, please write in the Telegram group and don't disturb the speaker. It breaks the momentum of the speaker as such. Okay, Nupur, I think we can.
there was one how to identify a different species as the views of externally. I think she asked in between of one of your slides. Kirti, can you unmute yourself and explain your question to sir? Kirti Yadav, are you there? Kriti Yadav, are you
Yes,
there?
ma'am.
Yeah,
Yes, ma'am.
please explain.
Ma'am, obey not. मैम जब ओबेरोनिया पजल जो था मैम, उसमें जो डिफरेंट स्पीसीज थे, तो एक्सटर्नली देखने पे मैम वो कैसे शो करेगा, मतलब कैसे आइडेंटिफाई करेंगे कैसे कि यही स्पीसीज है?
अर्जुन,
इस स्लाइड के बारे में बात कर रहे हैं?
यस सर।
बेसिकली पहले क्या होता था कि ओबेरोनिया के केस में स्पेसिफिकली जो नंबर ऑफ स्पॉट्स है, दिस इज नॉट अ कलर इमेज, ये कलर इमेज नहीं है। तो ओबेरोनिया के केस में जो फ्लावर का कलर है और जो फ्लावर पे नंबर ऑफ स्पॉट्स है, ये स्पॉट्स मत देखिएगा, बिकॉज ये स्पॉट्स वो हैं जो ब्लैक और व्हाइट में दिखाई दे रहे हैं, बट और लाइटर स्पॉट्स भी हैं, जैसे सी में है, तो बहुत तरीके के स्पॉट्स। तो उन कलर्ड स्पॉट्स के हिसाब से और उन कलर फ्लावर कलर के हिसाब से स्पीशीज जो है पहले डिलीनेट होती थी, बट काफी टाइम ये देखा गया उनका डीएनए निकाल के और उनके सीक्वेंसेस को, स्पेसिफिक रीजन्स को, जैसे यूनिवर्सल प्राइमर्स हैं या एसएसआर प्राइमर्स हैं, तो उनके रीजन्स को एम्पलीफाई करके देखा गया कि मोर और लेस ये डिफरेंट नहीं है, ये बहुत क्लोज है। तो ये या तो इकोटाइप्स हैं या मॉर्फोटाइप्स हैं। So there was a need to identify one or two important morphological features जो प्लांट को और जो एक स्पीशीज को दूसरे से अलग किए जा सके। और अल्टीमेटली ओबेरोनिया के केस में वो ये डिप्रेशंस और डिफरेंट ये जो जैगर्ड नेचर ऑफ लिप है, इस पेटल को लिप बोलते हैं ऑर्किडेसी में, ये जो जैगर्ड पैटर्न है लिप का ओबेरोनिया में, वो एक डिफाइनिंग फीचर बनेगा। और दिस वाज ट्रू एंड दिस वाज नॉट जस्ट, मतलब ये कोई पहली बार में नहीं पता चल गया था इनको कि लिप के पैटर्न से बताएंगे। इन्होंने बहुत जगह कोशिश की, पहले अपर पेटल्स और सेपल्स में देखा, स्पर में देखा, पॉलिनिया में देखा, एंथर कैप में देखा, बट उनके भी रिलेशन से जो डिफरेंसेस मिल रहे थे वो मॉलिक्यूलर वर्जन से अलग नहीं थे। बट जब ये कंपैरिजंस इनके साथ में बनाए गए, जैगर्ड नेचर के साथ में, जो मार्जिन्स के साथ में, इवन इन केस ऑफ ओबेरोनिया म्यूक्रिनेटा एंड ओबेरोनिया बोपन्ने, ये जो प्राइमरी प्रोजेक्शंस है और इसमें जो सेकेंडरी और टर्शरी प्रोजेक्शंस है, ये डिफाइनिंग फीचर्स बने इंस्टेड ऑफ कलर और एंथर कैप शेप और पॉलिनिया कैप शेप या फिर स्पर लेंथ में डिफरेंस। बेटा आपका आंसर क्लियर हुआ?
यस सर, थैंक यू सर।
thank you, Mr. Arjun. There is one more question from Kajal. She says, sir, the difference in the depression can be due to mutation, right? Then why different species for that? Then why different species for that? Because in Rhamnaceae. I think this is what the question is, right, Kajal?
As far as I can understand, she's saying that some plants may have differences, maybe because of mutation. Again, I would like to say that let's not jump to conclusions and say कि mutation ही होता है। जैसे कि मैंने इसके एग्जाम्पल में भी बोला, मॉर्फोटाइप्स और इकोटाइप्स जो हैं वो एक बहुत बड़ी चीज़ है। और यू नो बिना प्रॉपर एनालिसिस के ये कहना कि वो म्यूटेशंस हैं, I don't know, I'm not so sure what the question really was, but I think she's saying that depression.
No, actually, yeah, I left out a portion of the question. Sorry, Mr. Arjun. It is, she has completed the question once again. She has written that why is it so for different species? Because in Rhamnaceae family, Ziziphus jujuba plant differ due to mutation, but the same species are the same. But the species are the same. May you please explain, sir? Kajal?
We can ask Kajal to unmute.
Yeah, Kajal, can you please unmute yourself? Please explain.
Yes, ma'am. Ma'am, our teacher in college have told me about this. Like when she was teaching us the Rhamnaceae family, she told us that Ziziphus jujuba have many differences in plants, but the species are the same. One research student under her did a research on Rhamnaceae family, Ziziphus genus, and she took different plants, like 10-20 plants. She did the research, but when she gave her report, the university told her that the plant, the species are the same, so her report was rejected.
Yeah. So you want to know why or how we in a single species?
Yes, sir. Like there is difference in the morphological characters, but the species are the same. Why is that, sir?
Uh, this is a question which is quite interesting, and um, आप कभी अगर सोचें इसके बारे में तो आई डोंट नो वेदर हाउ यू हैव थॉट अबाउट दिस और नॉट कि एक स्पीशीज से एक नई स्पीशीज इवॉल्व कैसे होती है, एक एंसेस्टर से दो स्पीशीज कैसे बनती है ओवर अ कोर्स ऑफ टाइम. तो बेसिकली जो आप मुझे एक्सप्लेन कर रहे हैं वो ये है कि द स्पीशीज इज सेम बट दे आर डिफरेंट मॉर्फोटाइप्स और दे मे बी डिफरेंट इकोटाइप्स. तो बेसिकली धीरे धीरे जब ये डिलिनेशन होता है ना, जब एक ही स्पीशीज अलग अलग मॉर्फोलॉजिकल स्ट्रक्चर्स में प्रेजेंट होती है अलग अलग साइट्स में, फिर धीरे धीरे उसका अट्रैक्शन जो है, दे अट्रैक्ट डिफरेंट काइंड्स ऑफ पॉलिनेटर्स. जैसे ए साइट ए पे अगर कोई प्लांट है, उसका स्पर लेंथ थोड़ा छोटा है और साइट बी में जो है उसका स्पर लेंथ थोड़ा बड़ा है, तो वो डिफरेंट मॉथ स्पीशीज को अट्रैक्ट करेगा. और धीरे धीरे जब ये डिफरेंट मॉथ स्पीशीज को अट्रैक्ट करेगा तो पॉलिनेटर स्पेसिफिक हो जाएगा प्लांट. तो जो ब्रीडिंग होगी वो भी सिलेक्टिव हो जाएगी प्लांट की, वो अपनी ही पॉपुलेशन में होगी या सिमिलर पॉपुलेशंस में होगी. तो धीरे धीरे इट मे गिव राइज टू अ डिफरेंट स्पीशीज, बट दिस इज अ वेरी 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 लॉन्ग प्रोसेस. एज ऑफ नाउ आई कैन ओनली से कि जब तक रिप्रोडक्टिव बैरियर्स नहीं बनते हैं तब तक वो एक नई स्पीशीज नहीं है. वो जो उन्होंने देखे होंगे फीचर्स, वो एक बैरियर नहीं बन रहा है. मतलब मैं आपको मॉर्फोलॉजिकली एक्सप्लेन कर रहा हूँ, मॉलिक्यूलर एनालिसिस करके तो पता चल ही जाएगा कि दो स्पीशीज अलग है. बट आपने स्पीशीज कॉन्सेप्ट आई होप पढ़ा होगा. एक होता है मॉर्फोलॉजिकल स्पीशीज कॉन्सेप्ट, एक होता है जीनियोलॉजिकल स्पीशीज कॉन्सेप्ट, एक होता है रिप्रोडक्टिव स्पीशीज कॉन्सेप्ट. तो बेसिकली ये जो कॉन्सेप्ट है स्पीशीज के, इनमें बैरियर्स है. जब कोई प्लांट जो है इसमें रिप्रोडक्टिव बैरियर आ जाएगा और जब वो धीरे धीरे मॉडिफाई होता जाएगा तब वो एक न्यू स्पीशीज बनेगा. बट एज ऑफ नाउ जो चेंजेस, जो स्मॉल डिप्रेशंस हैं, वो इतने मीनिंगफुल नहीं होंगे उस प्लांट के एवोल्यूशन के लिए. और हो सकता है जो म्यूटेशंस होते भी हैं ना, म्यूटेशंस जो है कॉन्स्टेंटली प्लांट में होते रहते हैं और किसी एक पॉपुलेशन से स्पेसिफिक नहीं होते, जरूरी नहीं है. एक विद इन अ प्लांट पॉपुलेशन भी म्यूटेशंस हो सकते हैं. बट म्यूटेशंस प्लांट के लिए बेनिफिशियल है कि नहीं है, उस बेसिस पे वो सिलेक्ट होते हैं. और जब उनका सिलेक्शन प्लांट के लिए इतना बेनिफिशियल हो जाता है तब it gives rise to a different species. But these depressions may not be so meaningful for the plant. And the plant has not decided to delineate itself. And this must be reflected in the molecular analysis of their study, in the amplified regions of the universal spacers. Beta, kuch to I hope.
Yes, sir. Samjha. Thank you, sir.
Beta, you're from which place, university, and year?
Mithibai College, Mumbai University.
Which year?
I'm a graduate third year.
Okay. Yes. So I think sir very rightly explained all this, and you read about phenotypic plasticity as sir mentioned. Okay, so mutation may happen at one region. Suppose one plant is affected by some mutagen, and is getting mutated, that doesn't mean a new species is coming up, right there only. Until
Okay, ma'am.
unless it
Okay.
becomes a population and then gets reproductively isolated, establishes itself. So it's a long process which happens. So just one plant having a different phenology, that doesn't mean. So even your some of your students have, tried to explain it in their language. So very nicely explained, Arjun, this thing. And I hope, Kajal, you're satisfied.
Yes, ma'am. Thank you.
Okay. And then I think, no, but I think there
Okay,
is
there is
another.
one more question, Mr. Arjun. What is the best microscope to study pollen? I repeat myself, what is the best microscope to study pollen?
Pollen grains are extremely small structures. You can obviously see them in your normal binoculars also. When you do acetylysis, these structures of pollen grains are also visible in the normal bright field microscope. But the detailed, exine patterns can only be viewed in scanning electron microscope. It all depends on how much, where you want to put a stop to your question, whether you are satisfied with the results of acetylysis, viewing it in the bright field microscope, or whether you want to go in great detail and identify the different patterns of exine, then you can go for scanning electron microscope. Depends on the research question that you want to ask.
Yeah. Do you want to add anything extra in this question? If you want, you can always unmute yourself, Peter. Okay, there is one more interesting question from Kirti. That is, sir, pollen grains can cause allergy in some people. So, what could be the reason for this? What is the compound or chemical substance found in pollen that can cause this to happen? If you could tell any example.
The pollen grains do not release any compounds from within themselves. They cause a reaction within the body, and the chemical substances are released within the body. So, one of the studies is release of serotonin, which is otherwise found in normal, concentrations within the body. It causes the feeling of happiness along with endorphins. But release of serotonins in the respiratory tract causes itching. And pollen grains, when they are inhaled, certain people, that is why it does not happen to everyone. So someone might have a dust allergy, and you might not. So that is because the pollen grains which are in the air are causing an allergic reaction for that person. And you might also be inhaling the same pollen grains. The chemicals are not secreted from within the pollen grains. They induce a reaction for some people who are more prone to allergies. So these pollen grains, when they rub against your respiratory tract, uh, for some people again, they might induce the secretion of serotonin and other chemical compounds from within the body. So it is an immune response of the body and not a chemical secretion from within the pollen grain. I hope this clears it up.
I think this will answer her well. Uh, there are two questions coming from same person, Puneet Arjun. That is, what magnification do you need to see pollen grains? How can I measure the size of a pollen grain?
Okay. So basically, when you go for a scanning electron microscope, uh, I hope my slide is still visible in the background.
Yeah, yeah, it is there.
So the microscope itself, it provides a scale bar. And to answer your second question first, the scale bar be used for measuring the size of the pollen grains. To answer your first question, what magnification is helpful in studying pollen grains depends on what species of pollen grain you are studying. See, for example, the pollen grains of Malvaceae family are quite large, and if you closely look at a hibiscus flower, I'm sure you would still be able to see most of the pollen grains using a macro lens on a simple DSLR camera. So depends on the size of the pollen grain and the species that you want to study. Generally speaking, for members of Solanaceae and Asteraceae, usually in a scanning electron microscope, it is at I think at 3000x. I hope I've answered you.
Is it all right, Punit? You want to ask something more? Movement of pollen grains. What do you want to ask? Punit, can you unmute yourself, please?
Yes, ma'am.
Yeah. What do you mean by movement of pollen grains? You want to measure that?
Yeah,
Uh, Movement of pollen grains, as in from uh, one plant to another.
yeah,
I think he's written two things. How can I measure the size of a pollen grain? That is one thing.
I think I've
Yeah,
answered
that
the
he
size of the
has
pollen grain.
answered. We needed the
Yes,
movement of pollen grains.
and even if you that is, as he rightly said, with the microscopes you get a scale bar. Otherwise, as you would have done experiments, you use the stage and ocular micrometer.
Yeah, yeah.
Yeah,
You can
that
do
is
that,
also
and
used.
then you can, or otherwise, you have a third option of where, in cases where you don't get a scale bar, there is a software known as Image F, and you can just, uh, from the previously calibrated calculations from within the software, you can, drag a line across the area where you want to measure, and then you'll be able to get uh, the measurements of any particular structure, not just pollen grains. Uh, but that is a calibrated software. Otherwise, as Divya ma'am says, you can do it using the simple stage and ocular microscope.
Yes, because
And
whenever you have to take measurements under the microscope for student level and even for research purpose, if you're not having the software, as sir said, then always stage and ocular micrometer is used for measurements at the micro level. Okay, not below that. And I think, Arjun, there's a bombardment of question. Vishnupur, go
Yeah,
ahead.
right now we have many questions in front of us.
yeah, I would like to go through them one by
Vishnupur, I
one.
think you
I don't know.
missed the Kanupriya's question
Yeah,
also.
I will just pick up the questions that are left. So, Kanupriya, she asked, what is the function of spines in pollen grains?
Okay, the spines on the pollen grains, um, they are used by the plant, or they are a feature for the plant. So I'm sure you must have studied that some species have a wet type of stigma and some have dry type stigma. So these spines or these structural patterns on the exine are basically for the adherence of pollen grains on different types of stigma. So for a wet type stigma, you have exudates, stigmatic exudates, and pollen grains can easily be caught. So in such cases, in such plants where the stigma is wet, usually the plant has a reticulate or a smooth type of pattern. And in cases where we have dry type stigma, generally speaking, we have spiny or tuberculate patterns.
Yeah, I think that answers well. Uh, there is one question from Sapna. What is the best protocol to mount the dried pollens on microscope slides?
Best protocol for mounting, as
Mounting? Yeah.
yeah, what do you want to see? You want to test the viability, fertility, or you want to see the structures? So if you want to see the viability and fertility, you have basically the TTC tests and the acetocarmine test. So you can just mount it in the uh, uh, chemical itself. So in the TTC, you just mount it on the TTC solution. And in case of acetocarbene, again, you just mount it on the acetocarbene. If you want to do it if you want to study a pollen grain for scanning electron microscopy, you have a protocol. So you have an ethanol drying series, acetone drying series. Then you have a process which is known as critical point drying, and the third step is doing a sputter coating analysis. Sputter coating, where the gold particles are then coated on the pollen grains, and then the fourth step is viewing under the scanning electron microscope. So so it all depends on what do you want to see. It is all relative to your objective. So if you want to, study something in under a normal microscope, the protocol is going to be different. If you want to see it under scanning electron microscope, it is going to be different. If you want to test it for its viability, fertility, or germination, it is going to be different. So it is all relative, and the protocols are easily available on the internet. And I'm sure if you are going ahead with research, the protocols will most likely be shared by your superiors, or you're going to learn them sooner or later in masters or in your bachelor studies, I assume.
That's very true. Sooner or later, you will get one of the protocols that will be followed in your lab. So, Mr. Arjun, there was one slide in the beginning when you showed 3 different microscopes. I think from there, one question came up. Anna asked, do we use the corner right microscope from the last slide in pathology? I think that is from the first slide itself. She asked, do we use the corner right microscope in pathology? Am I right, Anna?
Basically, these are for, I just wanted to say that the one in the middle and the left tell you about the outer structures. They don't, cut through them. And the third one on the right, basically it's used for anatomical studies. Again, not just in taxonomy, it will be used everywhere. In pathological studies, in anatomical studies, in fungal, microbiological, anywhere.
Yes, even in your
Just wanted to show
diagnostic
that these are the
centers.
3 types of microscopes used.
Yes, even in your diagnostic centers, that microscope is there to
Yeah,
see, count the WBCs and see
yeah.
RBCs and see if the shape of the RBCs is fine, like in sickle cell anemia. So it's used for pathology as well. So these are just basic instruments you can apply to any field which requires the microscope.
Yeah.
Rightly said. There is one question from Ayush Arjun, that is, if 2-3 fruits are surgically jointed and they start growing jointly in that manner, can we say we surgically mutated that tree life?
Surgically.
I repeat, if 2-3 fruits are surgically jointed and they start growing jointly in that manner, can we say we surgically mutated that tree life?
You mean you're doing this uh, on the fruit which is on the tree, or you have excised it from the tree?
Ayush, can you please explain? Ayush, are you there? Yes, better.
Hello, sir. If we, sir, आवाज आ रही है?
Yeah, it is. You are audible. Can you please explain?
सर मेरा क्वेश्चन ये है कि अगर 2-3 फ्रूट्स अगर हम डिफरेंट प्लांट्स से, जैसे कभी कभी होता है एक ट्रांसफॉर्म ट्री बनाते हैं लोग फ्रूट्स की या फ्लावर्स की, जिसमें तरह तरह की अलग अलग जाति की फ्रूट एक ही उसमें लाइफ में जी रही होती है, तो क्या हम उसे ये कह सकते हैं सर्जिकली म्यूटेटेड हमने कर दिया उस प्लांट लाइफ की जो वैसी ग्रो कर रही है, जो मतलब अलग अलग वे में जीती थी अब वो एक में जी रही है?
नहीं, ये तो आप ग्राफ्टिंग की बात कर रहे हैं।
Yes, sir.
क्या है कि नेचुरली तो अकर करेगा नहीं, और आप ग्राफ्ट करेंगे तो आपको अगली बार रिजल्ट करने के लिए आपको कहीं और भी ग्राफ्ट करना पड़ेगा। तो नेचुरली तो आपको ऐसी पॉपुलेशंस नहीं मिलेंगी जहाँ पे 3 स्पीशीज जो है एक में ग्रो कर रही हो। एंड सेकेंड ऑफ ऑल, ये भी बहुत इंपॉर्टेंट है वेदर दैट प्लांट एक्सेप्ट्स इट और नॉट। जैसे कि मान लीजिए आपको किडनी की जरूरत है, अब आपको किसी ने किडनी दी, तो वो किडनी आपकी बॉडी एक्सेप्ट करेगी या रिजेक्ट करेगी, ये भी एक चांस है। ऐसे ही जब आप ग्राफ्टिंग करते हैं, इसलिए वी डोंट हैव सो मेनी परम्यूटेशंस एंड कॉम्बिनेशंस फॉर ईच प्लांट। प्लांट्स में भी सेम है, आप ग्राफ्टिंग करते हैं तो प्लांट कई बार रिजेक्ट भी कर देता है, और वो जो एक्सप्लांट आप लगाते हैं वो उस पर ग्रो नहीं करता, और ग्रो कर भी सकता है। और ग्रो करने पे हाँ, देर आर वीडियोज, देर सेवरल वीडियोज एंड सेवरल इमेजेस वेयर यू नो एक ब्रांच में लेमन निकल रहा है और दूसरे
Yes,
ब्रांच
yes.
में ऑरेंज निकल रहा है, but again those are very closely related species, both belong to citrus, and this all depends on whether कितना सिमिलैरिटी है प्लांट की। जो मान लीजिए आपने जिस प्लांट को ग्राफ्ट किया है तो उसकी रिक्वायरमेंट कैसी है। हो सकता है जो पेरेंट प्लांट है वो ड्रॉट पसंद करता हो, कम पानी पसंद करता हो। जो आपने दूसरा लगाया है वो ज़्यादा पानी पसंद करता हो, तो वो तो इतना खींच नहीं पाएगा। So it all depends on the combination that you are using. But surgically, मतलब joint करना, मतलब I think you are talking about grafting. Can we say surgically mutate that? No, this is not a mutation of life.
This is not a mutation. Exactly.
Yes.
I think Ayush, you have been answered well by sir. There is one more question from Shubham. He asks how MAT-K analysis is done, and do we have to do molecular analysis every time we are unsure about the species identification?
Molecular analysis or use of molecular tools for identification of species is more or less always used as a last resort, and because it is expensive. For that you need proper chemicals to isolate DNA using CTAB method, or even in some cases if you have secondary metabolites, you isolate them using a kit, which is expensive. You need primers, which again need to be made through a sequencing company, which come out to be expensive. You run it through a PCR machine, which is expensive. You run it through a gel and send it for sequencing, which is also expensive. So, because of the nature of the study and because of the costs which are implied within molecular analysis in taxonomic uh, delineation and identification is more or less always reserved as a last resort. And uh, that is how we have taxonomists who work on specific plant families or specific plant taxas. So you have experts in Nymphaceae, you have experts in Asteraceae, in Fabaceae. uh, Certain times you have experts in specific genres, like you have experts in Crotalaria. You have expert in your own college, in Shivaji College. You have Dev sir, who is an expert in Indigofera. So uh, an easier option is to approach an expert and use their expertise for identification of small detailed morphological features which can be help in delimiting or delineation of two species or two different plants. Uh, to answer how MATK analysis is done, MATK is a maturase enzyme within the chloroplast, and so it is basically a sequence, uh, and this is a universal marker. And again, as I explained, you need to have primers for MATK, and this region is amplified using PCR. You run it over a gel, and the region which is then expressed comes out in form of a band. You take out that band from within the gel and you send it for sequencing. You get the results of that sequencing, and then you have a uh, uh, MATK sequence for that particular species. I hope I really tried to answer your question. I went for the second one before the first, but I think it still does its job.
Shubham, are you? I hope you are satisfied with the answer. I think Sir answered very well, stating that molecular analysis is the last thing that comes to the mind because not every lab has that much fund. Not every lab is that rich, which I kept on mentioning in last to last lecture as well. So in that scenario, we cannot afford going for molecular analysis every time we are unsure about a species identification, right? Mr. Arjun, there is one question from Navroz. Uh, the question is, in one particular species, there could be more than one different type of pollen, like convex tricolpate and concave tricolpate pollens. So how to do nomenclature based on shapes of pollens?
Yes, we do have some species where more than two types of pollen grains are present. Uh, I don't understand by what do you mean by what you mean by nomenclature of pollen grains, but I think you have answered your own question, saying that one is a convex tricolpate, the other is a concave tricolpate, and uh, that is how nomenclature is done of the pollen grains. And to answer your first question, yes, there are plants which have more than two types of or more than one type of pollen grains. Also, in cases of some members of Proteus trimaceae, where you might have monads along with the dyads. But just to talk about nomenclature, I think you answered your own question.
Yes, and you have a NPC system of classification
Yeah.
based on number, position, and character
And character.
of the apertures. So all these are taken into account when you do nomenclature, or differentiate between the types of aperture the pollen grain have. So you please check on the net. You'll get about information related to that NPC system of pollen classification. So you have a chart there.
Rightly said. I think there are two more questions. One is
I
from
hope Arjun, you're not tired.
Yeah, that is
Not at all.
okay. That's good. We appreciate your time. Literally, we do that. There is one more question from Urvashi. She asks that, Sir, can we artificially create the pollen grains of the plants that are extinct now, like from calamities, and then can we regrow them?
Um, you can't artificially create a pollen grain, but you can discover them. So, as in many cases, pollen grains are sometimes, you know, uh, they are found as fossils, as I showed. Some people have tried to revive them by growing them in certain media to see whether they germinate or not. But uh, to grow a plant, you would still need a stigma to make a seed. Or sorry, not a gynecium to make a seed. Otherwise, you can have, and there are cases, and you have Divya ma'am, who is an expert in plant tissue culture. She knows about anther culture, where tissues can be used for making the calluses, but it all depends on uh, the nature and duration of the calamity, because the pollen grain may or may not have its viability still with it. So if a pollen grain has lost its viability and fertility over the period of calamity, it would be impossible to grow a tissue out of it. Just from itself, pollen grain, I don't think would be uh, could be used to artificially create a plant.
Very well answered, Arjun. There is one question again from Puneet. How does pollen grain develop?
Okay, so this is a big cycle. I'm sure you must have read about it, or must have must be going to read about it, because I think this is a very basic question in the reproductive biology.
Exactly.
Yeah. So you have,
Puneet, do you have anything in mind specifically before asking this question? Means, do you really want to discuss the development of pollen grain?
because then I can go into pollen mother cell, which then divides into tetrads and then has a meiotic division. But I'm sure you must have read this inside the book.
Yes, Malab, what do you mean by development? Development means in different manner. What specifically you want to know?
Puneet, can you please explain?
Yes, ma'am. हेलो
जी
Yes,
बोलो.
yes, better.
मैम वो माइक्रोस्पोरेंजियम के बारे में बता देते हैं
क्या माइक्रोस्पोरेंजियम के बारे में क्या?
इसका डेवलपमेंट कैसे होता है
किस पर्टिकुलर केस में, या आप जनरली पूछना चाह रहे हो? कैसे? You would have read about the processes of microsporogenesis and microgametogenesis. आप एक्सटर्नल डेवलपमेंट पूछ रहे हो, इंटरनल डेवलपमेंट पूछ रहे हो? एक्सटर्नल डेवलपमेंट जैसे सेम में दिखाया था, अलग अलग, वो अलग चीज होती है. आप पोलन ग्रेन्स का इंटरनल किस हिसाब से, आप क्या पूछना चाह रहे हो कि फीचर्स कैसे आते हैं?
इंटरनल बता दीजिए
Internal, so you have the processes of microsporogenesis and microgametogenesis, wherein you have meiotic divisions followed by mitotic divisions, right? So microspore mother cell forms your microspore tetrads, which get released into uninucleate microspores, then divide mitotically to form binucleate pollen grains, or then the second that is vegetative cell and generative cell, and again generative cell also divides to form your two sperm cells. This you were asking?
ये तो कोर्स में होगा.
हाँ, तभी मैं इसीलिए I spoke, because this is out of the relevance of the topic which you are
I'm sure,
taking up.
but you still would have been in, not taught this,
Yes,
or
this
you're
is
going
this
to
is
be.
in the course. पुनीत बेटा, आप यही पूछ रहे हो ना?
यस मैम
हाँ, तो वो तो चलो खैर वो अलग प्रोसीजर है, वो तो इस टॉक से नहीं है. बट यू हैव द प्रोसेस ऑफ माइक्रोस्पोरोजेनेसिस एंड माइक्रोगेमिटोजेनेसिस, जिसमें मियोटिक और माइटोटिक डिविजन्स होती हैं, जिससे आपका फाइनली पॉलन ग्रेन बनता है. जब यूनिन्यूक्लिएट माइक्रोस्पोर रिलीज होता है, उसमें एक्साइन पैटर्निंग नहीं होती है शुरू में. slowly and slowly sporopollen and is gets deposited on the surface, and followed by deposition of pollen kit material, and thereafter you have fully formed pollen grains, जिसमें पैटर्निंग जो सर ने सारी दिखाई वो विजिबल होती है अलग अलग, according to the different plant species. Sometimes it is species specific, right?
यस मैम
Okay.
Good job, Divya. I think Puneet has received his answer. In case, Puneet, you have certain queries related to the basic questions, you're most welcome to ask us anytime on Telegram as well, so that we can provide you material for the same. These are our course studies which are normally being conducted in botany honors in the 3 years of our studies. So I hope that you have got your answer. And there is one question for Arjun. Arjun, Govardhan has asked, sir, if any new species is found nowadays, beside the morphological characteristics, is molecular detailing also mandatory during the paper publishing of that new species?
Not mandatory, but people do it because nowadays people are interested in knowing where the species falls or which species it is more nearer to. So, as in the case of Oberonia uh, Bhupanne and Oberonia, uh, I'll just show you the slide again. as in the case of Oberonia boeppernii, which was later found out in this is the species on the right, and Oberonia mucronata, the orange one. Morphologically, you see that this species is more or less closer to mucronata. But on further molecular analysis, it was found that based on ITS and MATK sequences, that boeppernii is more closer to Oberonia gammei. And it is not necessary to, do a study on a molecular study to see to publish a new species, but yeah, people do it, but it is not absolutely necessary. It all depends on how far. That is how I keep saying to people, uh, आप क्वेश्चन पे फुल स्टॉप कहाँ लगाना चाहते हैं, वहीं आपका पेपर हो जाता है. आप चाहें तो पहले फुल स्टॉप लगा दें और जितना आपने किया है वहाँ पब्लिश करें, या आप डी पर जाएं और अच्छी जगह पब्लिश करें. दी क्वांटिटी एंड क्वालिटी of your data and research determines where you want to publish, but it does not determine whether it would be published.
Exactly. I would like to add that any kind of analysis, be it statistical or molecular analysis, supports your data. Your major work is done in the wet lab when you are identifying the particular plant. So if you want to support your data, you can go to any limits to create better, better measurements, better molecular analysis, statistical analysis. You can have a wonderful paper, but that doesn't mean that at the initial level you cannot get a paper published with just the basic studies. It's absolutely not a necessity, but yes, it can be a supportive ideology if you want to, right? There is one last question. I think this is the last question from Sumit. This is, although I have not studied plant taxonomy in detail, but I just try to correlate. Does apomictic species creates problem for plant taxonomists?
They are wonderful to study because they are a class apart. So basically, they are the exceptions to the general trend, and it is always very exciting to study the exceptions to see how they are different. But they certainly don't cause any problems. In fact, they are very interesting to study.
I hope, Sumit, you have received your answer.
No, I will just like to say something
Yeah, please.
to Urvashi.
Yes.
Urvashi, beta, you're there? 513 number. I really found this question very interesting.
Yes, ma'am. Yes,
level
ma'am.
that can be artificially create the pollen grains of the plants that are extinct now, like calamites, and regrow them. Can you think artificial, because ye bahut, at your level it is a very good question I found. What do you think artificial create करने के लिए pollen grain को क्या क्या चाहिए होगा, क्या क्या करना होगा?
मैम उस स्पीसीज के क्लोजली मतलब कोई जेनेटिक मटेरियल होना चाहिए और उसमें थोड़ा सा म्यूटेशन करके ताकि वो जो प्लांट मतलब जा चुका है या मतलब एक्सटिंक्ट हो चुका है उसके मतलब उस जैसे कैरेक्टर्स आ जाएं, इस तरह का हमें उसमें म्यूटेशन करना होगा कि उस तरह का उसके कैरेक्टर से अपीयर हो इस तरह से. एंड मैम उसको इन विट्रो हमें ग्रो करना पड़ेगा और मैम उसका जो है मतलब एपोगैमी इस टाइप से उसमें होनी चाहिए ताकि मतलब उसको ग्रो करने के लिए हमें या फर्टिलाइज करने के लिए हमें किसी और मतलब फीमेल प्लांट की जरूरत ना पड़े.
ओके, सो बेसिकली इन द सेंस इट्स लाइक कि तुम ट्रांसजेनिक क्रिएट करना चाह रहे हो.
यस मैम.
है ना, तो ट्रांसजेनिक क्रिएट करने के लिए अभी तक बिकॉज जो चीज़ें हम ट्रांसजेनिक्स में हाइपोथेसाइज करते हैं कि ऐसे करना होगा वैसे करना होगा, जैसे आपने पूरा प्रोसीजर बताया, एक्चुअली व्हेन यू प्रैक्टिकली डू दीज स्टेप्स इट ऑलवेज डजंट यील्ड रिजल्ट. ओके, तो जितना मैंने अभी तक पढ़ा है इस फील्ड में, बिकॉज आई कम फ्रॉम दैट फील्ड, दैट इज व्हाई आई एम सेइंग, सो अभी तक एक तो अगर आप जैसे सर ने बताया एग्जैक्टली कोई स्पीशीज एक्सटिंक्ट हो रही है या आपको फॉसिल मिल रहा है उसका, वो उस फॉसिल स्पीशीज की वायबिलिटी, पोलन वायबिलिटी अभी भी हो, दैट इज वेरी वेरी लेस चांस ऑफ दैट कि आप उसको कल्चर करो। हो सकता है, हो, ऐसा नहीं है कि नहीं हो सकता। तो वो
So मैम
डायरेक्ट
क्या हम उसको रिवाइव नहीं कर सकते हैं, मतलब किसी चीज से भी मतलब रिवाइव हो जाए, कोई ऐसी
रिवाइव करने
टेक्नोलॉजी
के
हो?
लिए जैसे आप लोगों ने अभी भी सुना होगा, जैसे आप एक न्यूबॉर्न बेबी जो बॉर्न हो रहा है उसका दे से ना दैट आप वो करा, इंश्योरेंस कराओ for the newborn, umbilical cord is
Stem
preserved,
cells. Yeah, yeah.
stem cell yes, stem cell what do you say, preservation.
यस मैम, यस
So
मैम।
for that those conditions would be required कि आपने उसको स्टैटिक रखा है। अगर एक टिश्यू मर गया है वो कैसे रिवाइव होगा? अगर टिश्यू एक पॉज्ड फेज में है कि उस पे, बिकॉज नॉर्मली ये जो प्रेजर्वेशन होता है -80 डिग्रीज पे करते हैं, दैट मीन्स वेरी लो टेम्परेचर वी स्टोर दैट टिश्यू फॉर एनी डीएनए एनालिसिस, आरएनए एनालिसिस एंड एवरीथिंग। ऐसा नहीं है, हम लोग हर्बेरियम से भी डीएनए एक्सट्रैक्ट करते हैं, लेकिन जनरली फॉर मॉलिक्यूलर वर्क इट इज प्रेफरेबल कि आप वो प्लांट मटेरियल को एकदम लो टेम्परेचर में रखो सो दैट इट्स लाइफ और एक्टिविटी इज हॉल्टेड और इट इज पॉज्ड बट इट इज नॉट डेड। समझ रहे हो? सो
मैम
फॉर
यस
एवरी
मैम।
प्लांट स्पीशीज द वाइबिलिटी एंड फर्टिलिटी वेरीज, कि हो सकता है कोई एक पर्टिकुलर स्पीशीज 1 साल तक वाइबल रह रहा है, आप उसको दोबारा 1 साल बाद भी कल्चर करने के लिए यूज कर सकते, राइट? तो वो
Ma'am,
अकॉर्ड, हाँ बेटा,
hello.
यस
Ma'am, but
बेटा,
we have heard the news of reviving a 30-year-old, 30,000 years old tree and 15,000-year-old tree. Ma'am, how is
But
that possible?
what happens?
these
You must have heard that those tissue they got themselves embedded in the
cold,
ice in the Antarctic,
Arctic, yes, cold
Arctic,
thing,
Arctic areas. These are
हाँ,
very, those are sub-zero temperatures.
so that is what I said. So fossil that time, fossilization is essential कि वो ice के अंदर fossilized हो।
Ma'am, then it is natural cryopreservation type.
yes 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 this is something which is happening like that naturally कि आप उसको प्रिजर्व हॉल्ट करके रख रहे हो एंड व्हेन यू आर गिविंग इट दैट द required conditions or optimum conditions for the growth, it is reviving back itself. Okay, so
मैम
that is, हाँ बोलो।
मैम वी कैन एक्चुअली वी कैन मतलब हम उनमें से डीएनए तो मैम एक्सट्रैक्ट कर ही सकते हैं, क्योंकि मैम जो कुछ चीज पुराने टाइम की
डेड
भी
में
कोई
से भी,
डेड
हाँ,
हो गई है
डेड में से भी कर
यस
सकते
मैम, तो
हो।
उसमें से एक्सट्रैक्ट तो कर
यस,
सकते हैं। तो देन मैम अगर हम उसको किसी न्यू प्लांट में या उसमें कल्चर करके उसमें म्यूटेशन लाके और मतलब उसके कैरेक्टर्स किसी और न्यू प्लांट में हम डाल
तो
सके, तो
नई
उस तरह से भी तो
स्पीशीज
एक
नहीं बनेगी, जिस प्लांट में डाल रहे हो उस प्लांट का ट्रांसजेनिक बनेगा।
ट्रांसफॉर्मेशन कर रहे हो
मैम।
आप,
समझ रहे हो?
आप उसको
यस
गिफ्ट दे रहे हो, लेकिन आप उसको एक फोर्स नहीं कर सकते कुछ नया बनने के लिए।
उसको एक नया ऑर्गेनिज्म नहीं बना रहे हो आप
Ma'am,
एक
she is basically talking about callus induction that could be
सो
carried out.
नहीं, दैट इज व्हाट फॉर एनी टिश्यू आप